All right. Hi, I'm Stephanie Gogarten from the University of Washington. My group is the coordinating center for some large human genetics consortia, and so we help manage data validation in those consortia. I'm specifically going to be talking today about the data model from the GREGoR consortium, which is a rare disease consortium, but we use the same validation process also in another consortium as well. And so if you're, trying to put together data from multiple different sources. This is a good method to make sure all of the data is ready to create a harmonized data set. So I'm going to go through some slides first, and then I'll give a demonstration of how the data validation workflows work. So I'm going to talk about how to define the data model, how uploaders prepare data to conform to the data model, how to validate those files against the data model, and then how to import them into an AnVIL workspace, and then we'll conclude with a demo. So first, what is a data model? So it defines what data should be present in an AnVIL workspace. And in AnVIL, we use the data tables feature to organize data. So we're not, storing like actual like genotype calls in these data tables, but we're storing file paths that point to data like BAM, CRAM, VCF files, as well as metadata about those files, so people can use them and have all the information they need to use them in an analysis. The GREGoR data model creates a database-like structure defining how the data tables relate to each other. And then the data model standardizes all of the columns in the data tables so it's easy to combine data across multiple workspaces. Uh, so as I said, we're going to be looking at the GREGoR data model for this demo. How do we actually use this in practice? GREGoR consists of five different research centers who are all generating and preparing data. And each of those centers is going to prepare data using the data model as a reference. They'll either just do that locally and then upload the data, or they can work directly in AnVIL in an interactive notebook. And once they have the data formatted in the data model, then they can save that to a Google bucket in tab separated value format, one file per table. And then they can run this data validation workflow using those TSV files as input. And it will generate a report in HTML format that'll tell them if there are any issues that need to be corrected in the data model before the data can be validated. And then if they need to make revisions, then they'll, work on that and rerun the workflow and iterate as many times as necessary until the workflow succeeds. And we'll take a look at that, what that report looks like in a few minutes. Uh, and then once the workflow runs without any errors, then they can set the import flag to true and import all of the data to the data tables in the AnVIL workspace. Um, how do we actually define this data model? So when uploaders are looking at the data model to prepare their data, they're usually looking at a Google Sheet. And I'll give you a quick look at what the GREGoR data model actually looks like. So we have a number of different tables. We have a participant table that has information about the participant in the study. We have a family table. We have a phenotype table where you can have multiple phenotypes for each participant. Um, and then once we get into the actual sequencing data, there's an analyte table, which refers to like a sample taken from a participant. There's an experiment table, which is the table that contains all of the details of the experiment that was run. And there's a different experiment table for every type of data. So GREGoR has a lot of data types. I'm not going to show all of them, but the one of those data tables is the experiment DNA short read table, and this has information that you might want to use in order to analyze whole genome sequencing data or whole exome sequencing data. Um, and then there's a table that has information about alignments. So this is where BAM and CRAM files are stored. There's an aligned set table, and this links the alignment files to a called variance table, which we have here. And so this shows you all of the VCF files that were generated from the sequencing data. So this is useful for uploaders to reference, but this is not useful for the data validation workflow to actually programmatically query the data table. So we also have a JSON format that's stored on GitHub, and this is like the canonical version of the data table. So this version of the data table is what the workflow actually reads and can compare the TSV files uploaded to. And so this is the format of the of that JSON file. We have the table name, whether the table is required, and then all of the columns of the table, and then information about each column. Is it the primary key? Is it required? A description of it. What's the data type that's expected? And then some examples and notes. All right. We also have a visualization where you can transform the JSON file into a format called the database markup language that generates a visualization of how all of the tables in the data model are connected to each other. All right. Um, so an uploader will, using this data model as their guide, create TSV files that conform to all of the tables that are expected in the data model where for which they have data. And then they upload those to the AnVIL workspace. And they any data files that are referenced in the data table, such as here's an example of a BAM file that's referenced in the aligned DNA short read data table. They'll have to upload those to the AnVIL workspace or to an AnVIL workspace. It doesn't have to be the same workspace. And then identify those files by the bucket path in the TSV files. And as I briefly mentioned before, we have a specific type of table called a set table, which is a built-in feature of AnVIL that can link multiple objects together. So the aligned DNA short read set table provides a link between the aligned read files, so all of the CRAM files or BAM files, and the genotype call sets, because each genotype call set is jointly called over multiple BAM or CRAM files. And so if we want to keep track of which BAM or CRAM files went into generating each VCF file. And so we have one column that's an aligned DNA short read ID that refers to each one of these IDs refers to a BAM or CRAM file. And then we have a set ID that is the same for all of these files and allows us to generate a set table in AnVIL. All right, so now I'm going to get into how the validation workflow is actually constructed. So we have a workflow that's called validate GREGoR model. And this workflow consists of multiple tasks. And one of those tasks is the validate data model workflow. And this workflow is a generic workflow that can work on any data model that's in the correct JSON format. So we use this workflow in other projects as well, in addition to GREGoR. And if you were to generate a data model in JSON format, you could use this workflow in your data as well. It's based on a Docker image that's hosted on Docker Hub by the UW Genetic Analysis Center called and the Docker image is called AnVIL util workflows. And this contains all of the code that the workflow needs to run. The Docker image has installed an R package called AnVIL Data Models, which is a package that I wrote that does all of the functionality of the data validation. And in turn, this R package is heavily based on two other R packages. One of them is called DM, which implements relational database logic in R. So all of the checks for like primary keys and foreign keys across tables are implemented using this package. And then all of the code that interacts with the AnVIL workspace is based on the bioconductor AnVIL package. So the AnVIL package has functions to interact with AnVIL data tables, so it can read from AnVIL data tables and it can write to AnVIL data tables. Um, It also has functions to interact with the Google Cloud buckets. So you can query the Google Cloud buckets. Note if you're using Azure, there's also functionality available for Azure. And so the AnVIL data models R package has a number of different functions to implement checks. So it can check that all required tables and all required columns are present. It checks that there aren't any missing values in the required columns. It checks that all of the data types match the column specifications. So if the column is specified as an integer, that all the values are compatible within an integer data type. If you specify enumerated values for your column, it checks that all of the values are one of those enumerated values. It can do conditional checks. So if you say you can have a column that's only required when another column has a specific value. It checks that all of the primary keys are unique. So this is a requirement of AnVIL data tables, is that each table has a column that is the primary key that's a unique value for that table. It checks that foreign keys exist in the parent table. So what that means is, say, the phenotype table refers to the participant ID. So it'll check that all of the values for participant ID in the phenotype table are also present in the participant table. And then it checks that all of the Google bucket paths exist, so that you're not pointing at a file that is not actually there. And then this package contains an R Markdown template that generates the data validation report. And then finally, it has functions to actually do the import of the data to AnVIL. And this package is available on GitHub. In addition to the sort of generic data model checks, the validate GREGoR model workflow also does a few consortium-specific checks. Um, for example, it checks that the MD5 sum listed in the data tables for each file matches the MD5 sum in the Google Cloud metadata. So one of the requirements in the GREGoR data model is that each file has an MD5 sum associated with it. And so this workflow will go and query the Google Cloud metadata without actually copying the file. So it's an inexpensive check to run and make sure that that MD5 sum matches. For VCF files, it'll check that the header of the VCF file matches the sample set in the data tables. Um, and you can also check that that the header of a BAM or CRAM file matches the sample ID in the data tables. Both of those checks actually require copying the file to instance and that the workflow is running in order to do that check, so they're more expensive. The BAM CRAM checks are especially expensive, so this one is turned off by default because it can be very expensive to run. And then it also, another consortium specific check is that the ontology terms in the phenotype table are formatted correctly. So the GREGoR data model phenotype table uses things like HPO terms to represent phenotypes, and so it makes sure that those conform to the expected format of ontology terms. All right, so once all of the data has been validated, then it can be imported to the workspace. And I'll just note that the validate data model workflow, so the inputs for this workflow are a map with the table names and the TSV files for each table. A map is a specific data type in WDL. The uh, data validation report is rendered, and then another thing that this workflow does is it writes output files that have some auto-generated columns. And I'll get to that in a second. And then if validation passes and the import flag is set to true, then the tables are imported to the AnVIL workspace. One convenience feature of this workflow is that if we have a table that doesn't have an obvious primary key, for example, the phenotype table has a participant ID to reference the participant, but a participant can have multiple phenotypes that are represented by the term ID. And so the primary key for this table is constructed out of both the participant ID and the term ID. So you need those two IDs combined in order to generate a unique value for this table. So what the validation workflow does is if the data model says that the primary key, the phenotype ID, comes from the participant ID and the term ID, it'll automatically generate this this I unique identifier so that the user doesn't have to generate that ID. It just gets generated automatically by the workflow. And so what that means is that the output of the workflow is going to is going to have a new TSV file that's actually what gets imported in the AnVIL data into the AnVIL data tables once that primary key has been added. So this is what the output of running the workflow will look like. And then each of these files that gets output from the workflow is imported to AnVIL. All right. So now we're at the demonstration. So I'm going to start on doc store because that is the um, place where the GREGoR data validation workflows are stored. Um, so if you go to the GREGoR consortium doc store page, there's a section for data validation. And this has a number of different workflows, many of which are component parts of the validate GREGoR data model workflow. But we're going to use the validate GREGoR data model workflow. So we'll click on that, and then we'll go to launch on AnVIL. And then I need to put in the workspace that we want to run this in. So I'm going to find my demo validation workspace. And I've already imported this workflow before, so we're just going to give it a new name to distinguish it from the previous import. And then we can import this workflow to the workspace. And so here we are noting that we're going to run multiple other workflows as part of this GREGoR data validation workflow. Um, so here is the all of the inputs for the workflow. So the model URL is the path to the JSON file. So if we go back to our GREGoR data model.json, we need to get the raw version of this. So you just get the plain text of the JSON. And so we would copy this URL and put it here into uh, our model URL path. I'll also note we're going to want to run workflow with inputs defined by file paths and not data tables. Uh, the table files are going to be a map type that have a string and a file type. And rather than typing all of these in, I'm going to go to my previously saved version of this workflow um, and see here are all the values entered. So the participant table is just participant and then a colon and then the path to the data table containing the data for the participant table and so on for all of the tables that we're going to import. And then we have the workspace name and the workspace namespace. So the workspace name is just the name of the workspace that we're working in, and the namespace is referring to the billing project for the workspace, which is here. And note that you don't have to actually run the validation workflow in the same workspace where you're actually importing the data. You can run it in a different workspace. Um, so you just have to make sure that in the workspace name and workspace namespace fields, these are where you want the data to actually be imported to. Then we have a number of flags where we can turn on and off different components of the workflow. So as I mentioned, the check BAM is off by default, and all of the rest of these are turned on by default, so they're true by default. Checking that the bucket paths exist in the for all the files that are in Google buckets, checking that the MD5 sums match, checking that the phenotype terms all have the correct formatting, and checking that the sample IDs in the VCF match the header and match the set table. Um, there's this column called hash ID nchar, and this is just referring to the number of characters that get generated in those automatically generated IDs that I talked about a minute ago. And you can generally just leave this as the default. Um, then we have the import tables flag. So usually when I'm running this, I'll start with it as false just to make sure that my validation works. I'll note that there are a couple of checks that aren't actually run until this is set to true. And those would be the check VCF and the check BAM, because those rely on actually reading data from the data tables after they've been imported to make sure that that the formatting matches, because it has to go through the relational database logic to pull out the correct identifiers. And so that happens after the data tables are imported. So if you set this to true, it's still possible that you might get a validation failure if you have one of those flags turned on. Overwrite is controlling whether or not you're going to overwrite existing data. So if you have data in your data tables already and you're trying to import data with the same primary keys, will it overwrite or not? This is false by default. So if you try to overwrite, it'll give you an error that says these rows already exist. And if you know you want to overwrite that data, you can set this to true. And then finally, the project ID is if you have a request or pays workspace where some of your Google Bucket paths are located, you'll need to have this project ID in order to be able to query the MD5 sums for those files. All right. So once we're all set with the workflow, with all of our inputs for the workflow, we can run this workflow and click launch. I'm not actually going to do that because it takes a little while. So I've already run the workflow in this workspace. So we're just going to go and look at the output from that workflow. So we can see that it failed. So we're going to figure out why did this workflow fail. So we're going to go into the job manager. And then we're going to look at the log file for this workflow to see why it failed. So if I scroll up to the error, it said table files not compatible with data model, see data model validation.html. So it's behaving exactly as we expected to, which is that if our data tables aren't compatible with the data model, it'll give us an error, and then we have to go and look at the report. So we'll go back to the job manager and click on the execution directory. And then we'll click on this data model validation.html. And this will give us a report. So here we can see that we have all the required tables, but we're missing some required columns. So there's two columns called solve status and missing variant case that weren't present in the data that I prepared. So the data that I used as an example here was created with version 1 of the GREGoR data model several years ago. And so the data model has evolved since then. So when I went to run the validation workflow again, we've added some new required columns to the participant table. And so this no longer passes validation. So if I would have to go back and make sure that I add those two columns to the data model in order to make sure that this passes. It tells us any optional columns that are missing. So there's quite a few optional columns that aren't present in my example data. It also tells you if there's any extra columns, which can be useful if you have a column that's just has a typo in the name. You might see that there's an extra column showing up, and you can compare that to the required and the optional columns just to make sure that you haven't you've given something the wrong name by accident. Um, so it tells us we don't have any missing values in required columns, which is good. All of our column types match the data model, so all of our integers are integers and so on. Um, and then it's telling us that there are some Google bucket paths that don't exist. So these files must have been moved since or deleted since I first set up this example data several years ago because I was using data from a public project with 1,000 genomes data. So it looks like all of these index files are no longer here. So if I actually wanted to get this to validate again, I would have to go and find the correct paths to the index files. So, and now we'll just scroll through a whole lot of index files that don't exist. until we get down to the end, and it tells us that all constraints are satisfied for primary keys. If we had any non-unique values for primary keys, that would show up here as failing this check. It also tells us that cross-table references are satisfied, and this is where it would tell us, say, if we had in the phenotype table we had a participant ID that did not exist in the participant table, that would show up here in this part of the check. Okay. If we wanted to get this to validate, we would have to fix those errors. We can also look at the job manager because this is the validation is one part of the workflow, but if we look at the list view of the workflow, we see that it also did something else, which is that it ran this validate MD5 task. And it runs one of those tasks for every data table that we provide is it checks all that each of these shards is checking the MD5 values in one data table. And we can see that all of those succeeded. So even though we had a couple of validation errors, we know that all of our MD5 sums in the data tables are correct. Once we've actually gotten the data to validate, um, we can see what it would look like in AnVIL after the data tables get imported. So here are all of those tables that saw in the data model earlier, and they're all present in the AnVIL workspace. So we have a participant table that has participant data. We have a family table. We have an analyte table that refers back to the participant ID. Um, we have an aligned DNA short read set table that has all of the CRAM files listed. And then here's the set table that I mentioned earlier. So it has a set ID, and then it refers to entities in the aligned DNA short read set table. And note that these are a subset of the total number of CRAM files. Not all of those CRAM files were part of the joint call set, so we only have 2,500 entities in the set table. And this called variance DNA short read file refers to this set ID, and then each of these per chromosome VCF files, we know the samples that in each of these VCF files are the samples that are in this set. Um, we can also note in this table, this is one of the tables that has an automatically generated ID. So the workflow generated these IDs, the primary key for this table. All right. So that concludes my demo, and I'll just go back to the slides and ask for questions, and also note that we have all of this is available on DocStore and on GitHub. Um, so you can look at this code and adapt it for your own use if you want to validate data in your own projects.